Okay, doctor, can we start? Because our Dr. Tan also already here. Can you hear my voice clearly?
Hi, Tasya. I cannot hear you.
How about now, doctor? Is it can you hear me? Can you hear me?
I cannot hear from you all. Sorry.
I can hear you, Tava.
Maybe, doctor, you wear the headset.
Mhm.
Maybe check the setting and the audio. Semester October 2024. This webinar is organized by Faculty of Applied Sciences, UiTM Perak Branch, Tapa Campus in collaboration with UiTM Sabah Branch, Kota Kinabalu Campus, and University Malaysia Pahang Al Sultan Abdullah, UMPSA. Thank you all for taking the time to join us today. My name is Mrs. Tavasurya Ilangovan from Faculty of Applied Sciences, UiTM Perak Branch, Tapa Campus, and I'm the host and first moderator for today's session. Together with me is the co-host and the second moderator. She will help me during this session. I would like to invite the second moderator to introduce herself. Dr. Shazwani.
Okay, Assalamualaikum and good evening, everyone. My name is Shazwani. I'm biology lecturer from UiTM Sabah. Nice to meet you all.
Okay, thank you, Dr. Nur Shazwani binti Mota. Next, I would like to introduce the team members of BIO122 Collaborative Teaching. There are 5 lecturers from Tapa campus and 1 lecturer from Sabah campus. Puan Wan Nurul Hidayah Wan Anwar, Puan Nadia Nisha Musa, Puan Zaidatul Shakila Muhammad Ashari, Puan Nur Hazira Azmi, and Dr. Shazwani binti Mota from Sabah campus. Thank you all for your support and cooperation for this collaborative teaching. Dear students, in this master class, we will share about the topic plant tissues. So stay tuned to learn more. Before the presentation begins, allow me to brief you on some house rules and how this session will run. To ensure the smoothness of this session, please mute your microphone when you are not speaking. We would like to request that all the students will remain in this room until the end of the session. Your cooperation is highly appreciated. The link of attendance will be shared at the end of the session. Please note that the attendance link is available for 30 minutes after the session is ended. Then the guest speaker will start to present after the moderator introduces the presenter. Firstly, the guest speaker will present the material for about 1 hour. After that, there will be a 10-minute break. After the break, the session will be continued with another 1 hour presentation by the speaker. Following that, we will have a small game using Quizlet and Q&A session with the speaker about 10 to 15 minutes. Dear students, feel free to drop your questions on the topic through the chat box, or you can unmute to ask the question along with your full name and affiliation. When asking a question, please turn on your webcam. Not only that, we also advise the students to on your camera while the session conducted. Any further questions after the stipulated time, we highly recommend you to discuss with the speaker via email or through the personal chat. Finally, the session will be ended with a challenge game session and conclusion. Also, don't worry about taking the notes. We will provide the handout and the session's recording video to your class lecturer within 3 days. Okay, before introduces the presenter, I would like to invite the guest speaker and all the participants to get ready for a photography session. Everyone, please turn on the camera for the photo session. So one more reminder, everyone, please turn on the camera for the photo session. Please make fast. So Dr. Shazwani, are you ready for the photo session?
Yeah, I'm ready. Okay. Anytime.
Yeah. So we wait for the students for a while, few minutes. Let them to on their camera. Actually, here 308 participants. So please make sure, please on your camera. We're going to snap photo. So we wait for another 1 more minute. We will take the photo at the beginning and at the end of the session. Okay. So no problem where you are. Okay, let's restart.
Yeah, we can
Three,
start now. I already snapped the pictures. Yeah.
okay. 3, 2, 1.
Okay.
So can turn one by one, right? There are many
Yeah, there are a few pages.
few pages, right? Okay, so once doctor finish, then just let me know. We continue.
Ya, on din niya. Guys, nakot gila line.
Kejap ya, kejap lagi. Banyak page ni sampai 10.
So far, we have 329 participants.
Yeah, I think I got all already. Alright. Yeah, we
So
can
we
start
proceed?
now.
Okay. Okay, thanks, Dr. Nushazwani. Without further ado, I wish to introduce our guest speaker, Associate Professor Dr. Tan Swat Hin from Faculty Industrial Sciences and Technology, University Malaysia Pahang Al Sultan Abdullah, UMPSA. In the year 2010, she graduated her doctoral degree in biotechnology from University Putra Malaysia, UPM. Previously, from the year 2018 until 2019, she had a professional membership in Malaysia Board of Technologies, MBOT. Then her area of expertise are plant tissue culture on herbs, crops, and ornamental plant, and plant secondary metabolites. Dear students, now allow me to welcome our speaker, Associate Professor Dr. Tan Swat Hien, to present about plant tissues. Okay, Doctor.
Hi, everybody. How are you?
I'm fine.
I'm glad to be invited by Miss Dawa for this sections lah. Okay. So where are you all from? From Tapa or from Sabah?
Tapa here.
Tapa here.
Tapa.
Sabah here.
Tapa here.
Tapa here.
Sabah.
Tapa.
Oh, a lot from Tapa. I heard that Miss Tawa said you got 4 different places, right?
Actually, Doctor, four classes from Sabah, around 96 or 98 students. And from Tapa, we have 480 students. So overall, we have 530 students should join for today's session.
Okay. I try to share my screen. Is it okay? Can see my screen sharing?
Yeah, can see, Doctor.
Okay. So I'm Dr. Tan from UMPSA. I'm very glad to be invited lah for this talk. So for these plant tissues. Okay. So I start. Okay. So students, you should know about at the end of these sections, you should able to state and define the terminologies in plant tissues lah. Then you should able to identify, draw, label, and describe the characteristic of plant tissues. I heard from your lecturers that these chapters will be covered in your final exam lah. So hopefully you all can pay attentions when I describe about some of the features so that you can answer well in the final exam, hopefully. Okay. Okay, students, plant organizations. So this is a part of plant organizations. It's a cellular organization level of plants. Do you know what is these are for the first pictures? Anyone?
Plant cell.
Yes, it's a plant cell. All right. How about the second one? You can type in the chat box as well if you want.
Leaf surface.
Tissue.
Yes, correct. Tissues. Very good. How come it's open already? Okay, the third one.
Organ.
Really? Sorry.
Okay.
How come it's disappear? Yeah, the third one is organ. I don't know why the fading got problem.
Or maybe Doctor try to click the slide view, the slideshow at the bottom. Okay, now, yeah, now can see.
Yeah. Okay. So third one is organ, yes. The fourth one will be the system, then it will become the organism itself lah, basically. Okay. So what is the functions and plant growth process? Anyone can guess?
To do photosynthesis.
Yes, for photosynthesis purposes. So process of photosynthesis. How about this one?
Nutrient absorption.
Yes, correct. Nutrient absorptions. Beside that, the process of growth is for what? What is this picture for?
Transpiration.
So this one is about the flowers. So we talk about the ovary. So it's for what?
Reproduction.
Yes, correct. Very good. It's for reproductions. How about this one? Can you guess?
Structural support.
Wow, structural support, yes. The last one is the transpirations lah. Okay. So basically, when we have plants, you should know that they have a shoot system and a root system lah. When I use pointer, can you all see the pointer of my pointing? Cannot.
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 Hmm.
So how? Is that what I just talk like that lah? Then they cannot follow where I point. I never. You
Maybe
unmute.
Doctor, try to click the annotate. Because here, right, I cannot
Or the pen
annotate.
and laser pointer tools.
Yes, yes. They got annotate on the top right.
Okay, then now can see?
Okay, yeah, can.
Okay, good. Okay, so when we have plants, okay, so you should have the above will be the shoot system lah. So below will be the root system. So roots and shoots form a system where they have organs. Then you have your roots, you have your stem, and you have your leaf. Okay, so you have your apical parts. So within the stem, they have internodes. They have the nodes here. And we have the apical parts. We have the leaf with plates and petiole and the taproots. Okay, and the lateral roots lah. So these will be the whole plant organizations. So what is the functions for the shoots? Anyone can guess?
Transportation of water.
Sorry, cannot hear. There are few functions for the shoots. First is of course for photosynthesis lah, because you have leaf. Then we for transport of foods of water inside the stem. For reproductions and the storage lah will be the fruits. How about the root system? Anyone? Why a plant should have a root?
Absorb nutrients.
Yes, for absorb nutrients, absorb water and minerals. Beside that,
To hold the plant in place.
yes, to hold the plant in place so that it can stand firm. Beside that,
How about transpiration?
transpiration. That's mean transport of food and water, is it?
Soil.
If
To also hold the ground in place.
yes. So with this will be some of the function lah. When you talk about hold the plant in the place, will be anchorage lah, absorption of water and mineral, transport of food and water, and reproductions. Okay. So when we talk about plant tissues, basically plant is a multicellular eukaryote lah, with a tissue system. They're made of various cell types that carry out specific functions. So basically, there are three main tissue system. We are called dermal, ground, and vascular. So what is the dermal tissue are? When you look at the leaf here, you cut it into half of it lah. You can see it. Okay, you cut it. So the cuticles and epidermis will be the dermal tissues lah, if you have the leaf. If you cut cross sections of the stem, so you can see the outer layer should be the epidermis. which is the dermal tissue as well lah. For the roots, if you cross sections of it, so you have the root hair and the epidermis as well, will be the dermal tissues. Okay? Then we have
Okay.
the ground tissues.
Okay.
So ground tissue will be the palisade mesophyll lah, which is inner here lah, okay? So inside there will have the vascular tissues formed with xylem and phloems, okay? So again with the spongy mesophyll lah, ground tissue as well. If in the stem, then you can see the ground tissues is in the cortex and the speech. The most inner side is the speech lah. Cortex will be the outer. And the vascular tissue, of course, is formed from xylem and phloem again. And we have one more, which is the vascular cambium. The ring, black ring here is the meristemic tissues lah. Whereas for the roots, same, they have the cortex, endodermis, the orange color, and the white color is the pericycles. This is ground tissues. And for the roots, the xylem and phloem is a little bit different. If you have stem, they are in a circle shape. The leaf also in a circle shape. But for the roots, it's in like a triangle shapes. Okay. So they are the phloem is the green, the outer one, and the xylem will be the inner one lah. So xylem and phloem will form vascular tissues, whereas cortex, endodermis, pericycle will form the ground tissues for the roots. Okay. So plant tissues, when the permanent tissues are tissue that lost the ability of divisions and are completely grown lah. So as I mentioned just now, there are 3 types of them. What are they?
Dermal, ground, and vascular.
Wow, good. No, you can score already. Okay. So dermal, ground, and vascular. Okay. So look at the function of them. There are dermal tissues at the outer layer, as I mentioned, okay, covering the whole plants. And there are ground tissue system, which is hard to carry out the photosynthesis and store the photosynthetic products and support the plants lah. Okay, this is the leaf lah functions. So you have the vascular tissue system in between the ground tissues to conduct the water and solute throughout the plants lah. Okay. So same for the stem, you have dermal, ground, and vascular. Okay, and roots as well. So in the shoots here, we have shoot apical meristem. So this is not the permanent tissue lah. So they still have the ability to divide. And for the root apical meristem as well. Okay. Okay, just to test you all, we have dermal tissue made of what component? The most outer dermal tissue, what we call it just now?
Epidermis.
Yes, correct. Epidermis. Another one?
Periderm.
Yes, periderm. They help to do the protection and prevent the loss of water lah, basically for the function of dermal. How about the ground tissues? What are the components are?
Parenchyma.
Parenchyma,
Cortex.
cortex,
Parenchyma.
okay.
Sclerenchyma.
Yes, correct. So they help to do storage and supports lah, and metabolism as well. How about the vascular?
Xylem and phloem.
Yes, correct. Xylem and phloem. What is the function for xylem and phloem?
Transport water and sugar.
Yes, transport water and sugar. So this is the summary of it,
Xylem.
basically. Yes. Xylem and phloem. Yes. For the vascular tissues, it's comprised of the xylem and phloem. Okay. How plant develop actually is from the tiny seeds lah. So it grown into a plantlets, then it grown into a tall plants, then it's fruitings lah. So this is how plant develops. In order for plant to develop, they have to have the meristem tissues. Okay, so meristems is the regions of the plant where composed of cells that can divide lah. Okay, so if in these pictures, then we can see it should be the apical meristem and the roots apical meristem lah. Beside that, they have another two kind of meristem we call intercalary meristem and the lateral meristem. Okay. So what are meristem tissue are actually? They are undifferentiated tissues. So it can be consists of a clump of the small cell with dense cytoplasm and proportionally large nucleus. They can undergo repeatedly cell division to retain the ability to divide by mitosis. So they have two types, as I mentioned, apicals and the laterals. Okay. So the function of apical meristem is to elongate the roots and the shoots lah, so it can grow taller and taller. Whereas the lateral meristem is to help to produce the increase of plant in circumference or give. What does it mean? That means it go fat, become bigger, fats and fats lah. Okay, in terms of human, we say is become fat and fat lah. Okay, easier to say like that. Okay, so it located at the tips of the shoots and roots lah, where we have apical meristem. So the shoots apical meristem is found at the tips here. So if you want to know where is the tips of the plants, then everyone eat cabbage before?
Yes.
Yes.
Yes. Do you have to peel the cabbage until the end of it?
Yes.
Yes.
So you can see a shape like this, right?
Yes.
So this basically will
Yes.
be the apical meristem lah, inside the cabbage lah. Is you have comprised of young leaf, prototerms, central mother cell, and the meristem, procambiums, nodes, axillary buds, and the pitch lah. Okay. So they are responsible for creating cells and enable primary growth lah. When we talk about primary growth, then we talk about growth in length to drive the plant into the light and air lah. That's why you can see the plant become taller and taller so that they can do photosynthesis and exchange build up gases lah. Whereas plant tissues that result from primary growth are called primary tissue, which make up primary plant body lah. Okay. When we have roots, meristem, then they comprise a few things here lah. What you can see from here, they comprise of what? In the root meristem, it comprises of root hair. You have the prototerms, ground meristem, and the most inner side is the procambium. So this prototerm, ground meristem, and procambium make up the primary meristem lah. Whereas the root apical meristem is found at the tips of the roots lah, and it covered by the root caps, which is to protect the root apical meristem. So when we have a root apical meristem, is to sense the condition of the soil around the root so that it can help to direct this plant towards the water and the desired nutrients lah. So can carry many of the nutrients as their needs, allow the root to extend into the soil. Okay. So in the apical meristem, they give rise of 3 types of primary meristem lah. So do you know what is the 3 types of? Anyone?
Protoderm,
Yes, protoderm. Another one?
ground meristem,
Ground meristem.
procambium,
Yes, procambium. So protoderms will be at here to form the epidermis lah cell, whereas you have a procambium here to form the primary vascular tissues, and you have a ground meristem, the inner side, to differentiate into the ground tissues lah, where is the permanent tissues formations lah. So as I mentioned just now, you have the intercalary meristem arise in the stem internodes here. Okay. So this is about the summary lah. You have an apical meristem. They will form 3 types of the primary meristem, which is the protoderms, ground meristem, and procambium. Where protoderm can form epidermis, which is the dermal tissue system, and ground meristem will form ground tissues, which you just now you mentioned already is a parenchyma, collenchyma, and sclerenchyma is forming ground tissue system, and procambium can form xylem and phloem, which is the vascular system. Okay, so this is just to remind you all. If you want to label it, you need to memorize this one. The protoderm, procambiums, apical meristem, round meristem, shoots, and vascular cambium and cord cambium to form the lateral meristem. So this is the, what is these pictures and this picture difference actually?
Zoom in.
This is a zoom in. This is the actual plant cell we taken under the microscope that stained with the color so that it can be visible by us lah. Okay, so it can be like this. This is the drawing. So this is the actual one. So you can see it's a leaf precambium, and you have the apical meristem. You have the axillary buds primordium. You have the ground meristem, procambium, and the protoderms. Where you have leaf arise from as a leaf precambium, there are singular and primordium. If you have axillary buds, then it can form from lateral shoots and some later time. So a little bit on this part as well. So this will be again from the real roots we stain on it and view under microscope. So the outer layer will have the protentum. Okay. So inner a little bit is the ground meristem, and the center will be the procambium. And you have the apical meristem of the roots. Okay. And you can see this one a little bit different from these pictures, right? So this will be the root cap in protecting the root elongations. Why we need the root cap in protection of the root elongations? Anyone can try? Why we need a root cap to protect the root elongations?
Because that is where the multiplication of cell happens.
Okay, because the soil is hard, right? So when they want to elongate, they have to penetrate in. So the root cap is to help to protect the epitome meristem of the roots lah, to do the divisions. Okay, so this one is for the pictures that for us to draw lah, if you want to draw of it. As I mentioned, you may need to draw, right? Okay, so you can draw like this lah. So this one is the vascular cylinders. Then you have the cortex. Then you have the outer layer will be the epidermis. Then you have the root hair. Any questions so far?
No.
No.
Okay, then we continue lah. So just now is about the apical meristem. So what is the function for apical meristem actually?
The differentiation of cell.
The differentiation of cell to help the cell to have a both also lateral meristem also have differentiation of cell. But why we call apical meristem and we have another one is lateral meristem?
Elongation.
It is for
One is to let the plant to grow.
apical is for the growth of plant.
Yes, to grow
Such as
high.
height.
Is to let the plant grow high, right? And the lateral meristem basically is to increase the diameters of the plants. So we call secondary growth lah. So basically for lateral meristem, we have two types of them. One is called vascular cambium. Another one is called Cook cambium. So this vascular cambium and the Cook cambium will form the lateral meristem lah. Other part will be the same. Okay. So in woody plant, the Cook cambium will produce the Cook cell and replace the epidermis with the thicker and tougher periderm. Whereas the vascular cambium will produce the secondary vascular tissue. You have a secondary xylem and secondary phloem lah. So in the next pictures, you can see when you have the early spring, they only have a primary growth lah. Okay. So with the epidermis, cortex, primary phloem, and the primary xylem, in between, you have the vascular cambium lah. After late summer, they start to grow laterally, okay, in diameters. So they will form the Cook and the Cook cambiums, and the secondary, the lighter color one is a secondary phloem lah, and the secondary xylem here. So same goes for the year 2 lah. They will grow like this. So the plant growth, it will result cell divisions in apical meristem at the shoot tips and resulting the growth of the plant's length as well. Okay, so we use the lateral meristem. So primary growth of the plants allow roots to extend throughout the soil and for the shoots to increase the exposure to the light and carbon dioxide lah. Whereas for the secondary growth, if for the plant, is the result from the cell divisions at the lateral meristem and increasing the shoot give and thicken the plant bodies lah, basically. So this is an overview of the primary and the secondary growth lah. So you can see they have a shoot apical meristem, where that's why when you pluck the plants, they still continue grow because it's at the they have the shoot meristem here. Okay. So in the woody plants, they have a lateral meristem and add on the secondary growth to increase the gift. Okay. So the vascular cambium and the cord cambium can form the lateral meristem. So this is the pictures of the primary growth and the secondary growth in the stem. So far, okay. Can you remember if I ask you to label this?
Not yet.
Inshallah can Madam.
Inshallah can, ah. Atau nak tutup sekejap, bagi your label. You want to label? Then I tutup sekejap. Okay. So same lah. You have your primary growth. So outer layer will be the epidermis. Then in between will be the cortex. Then we will have the phloem, primary phloem, and the primary xylem lah. So center with the speech. So this one, when they have secondary growth, they will have pyramidoms, cord cambiums. Okay. So inner side will be the same. And we have the secondary phloem and the secondary xylem lah. So inner will still remain the same lah, basically, as the primary growth in the stem. Okay, so organizations of primary tissues in stem, in most of the monocot stem, their vascular bundle are scattered throughout the ground tissue lah. So the ground tissue will be at the center, then you have the vascular bundles here, look like the small vacuoles, okay? So you have the vascular and then protected with the epidermis lah, okay? The membrane epidermis. Okay, can you give me some examples? What kind of the plants that have monocots actually? Monocot stem. Anyone can
There
guess?
Grass.
Yes, grass,
Bamboo.
maize,
Palm tree.
palm tree. Palm tree, yes.
Wheat.
Weeds, yes.
Onion.
Sorry?
Corn.
Corn. Yes, corn definitely is a monocot. How about banana? Is it monocot?
Yes.
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 We have bamboo as well. Then we have sugar canes. So all these have a monocot stem. They have a vascular bundles scattered around the ground tissues lah, basically. Okay. So we have another type of the stem we call eutychot stem lah. Okay. So this kind of the plants, they have vascular tissues. Okay, so this is the vascular tissues. Consist of the vascular bundle arranged in a ring. Just now is scattered. This one arranged in a ring. So if you can see, the phloem will be the blue color, and inner side we have the xylem. Then just now we have the ciliary cyma and the ground tissue connected to the pitch to the cortex. Okay, and this is the cortex. So again, you need to know where is dermal tissue, ground tissue, and vascular tissue lah. Anyone can give me some example of the eutychot plants?
Roses.
Sunflower.
Yes, sunflowers.
Roses and maple
Roses,
tree.
yes.
Tomato.
Tomato, yes. So some garden plants, shrubs and trees lah. Usually the broad-leafed flowering plants all will be under category of eudicote stems lah. Okay. Then we have organizations of primary tissues in young roots. So this is the roots lah in the cross sections of the under the microscope lah. You can see. So we have the outer layer. This one outer layer will be what? What is this called?
Epidermis.
Yes, epidermis. Then inner a little bit will be the
Cortex.
cortex. Then no center part.
Vascular cylinder.
Yes. So vascular cylinder is will have the form of two kind of tissues. What are they? In the vascular cylinder, we have two type of tissues, right? And I mentioned earlier. So what are they?
Xylem and
Xylem and
phloem.
phloem.
Yes, correct. Xylem and phloem. Okay. So this one is the cross sections of the monocot roots in the vascular cylinder. Okay, with the core parenchyma cell surrounded by ring by alternating the xylem and phloem lah. So this one is for the eudicots roots lah. So that's why you can see the arrangement is a little bit different as same happen in the stem as well. So for the lateral roots, we have arise from within the pericycles, okay? When we look at this one, the outermost cell layer in the vascular cylinder lah. So this is the imagined lateral root from the vascular cylinder. So basically, we have plant tissues. So plant tissue comprise of meristematic tissues. When they grow old, they will grow become the permanent tissues. So you have the dermal tissues, you have the ground tissues, and you have the vascular tissues. So dermal tissues comprise epidermis and peridermis, whereas the ground tissue comprise of parenchyma, collenchyma, and mesenchyma, and vascular will have the xylem and phloem. For apicals, it consists of primary growth and lateral for secondary growth. So this is a little bit on the summary of the first part lah. Okay, so let's join for Quizlet Live on the Meristem. Okay, I need to enter the. I stop sharing first. I need to enter the website to give you the code. Wait for me, Okay, so students, you can scan the codes and join the games. Can you able to scan it? Okay, yes, I saw few names. So we wait for others to join. I heard that you have 320 of students here. So I try few kind of games like Kahoots. They only can manage to have 10 students. So lastly, I found this Quizlets Live lah. So you can accommodate like 50 of you all at least. So I think everybody can try to do it lah. Okay, anyone else want to join more before we start the games? Mm. Okay, got 91. You can change your photos if you want the pictures. Okay, let's wait. Okay, already 152. Anyone still want to join? 158. Ooh, 160. Okay. Okay, let's wait for 1 more minutes for those who want to join. Then by 9 o'clock, we shall start the games now. Okay. Seem like nobody joining anymore. Then we shall start. Okay, you can start answering. You can see your friends is jumping in, become the champion. Okay, Glenn is getting 2 marks here. Wow. Wow, Shao Hill is matching. Oh, Sasa is matching. I mean, that's what I mean. Zuleika. Lulu. Paul also getting in front. The one then have half of it being corrected. Diva then still leading. Whoa. Tak lian lan ah.
Hai. Online juga di semoga ini juga.
Dr. Tan, can you hear me?
Yes.
Because here no more screen sharing.
Sorry?
Because here I didn't see any screen sharing. That's why I thought to ask.
Oh, it stopped already. Ah.
Yeah.
I share again. Wow, now we convener. Let me share again.
Okay.
Sorry, yeah. Because usually I use other programs. Okay, we have 3 winners. We have Indi, Bunga, and Daniel Ashraf. Take a quick clap for them to get the full marks. Wow. Congratulations. Okay, others work hard lah. Okay. Okay. So thank you for joining the games. We take a break for 10 minutes. So now is 9:08. When we come back by 9:18 and continue the class, you can go for toilet breaks or whatever. Okay?
Okay, doctor.
Okay, doctor.
Okay, can you see the slide?
Yes, can, doctor.
Nampak.
Nampak? Okay. Then we continue lah. The permanent tissues, the meristematic tissues, will divide and differentiate to form the, sorry, I use the laser pointer first, okay? Will differentiate to form the permanent tissue lah. So when you have a permanent tissues, then they will be completely grown and lost the ability to divide. So there are 3 types of tissues, which is the dermal, ground tissues, and the vascular tissues. So each of them make a tissues system, connect all the blood organs. Okay, so just now I mentioned the dermal tissues will comprise of what? This is what tissues? Can you guess? Anyone?
Epidermis.
Yes, correct. Epitomis. You are right. How about the bottom one? What is this one?
Stoma.
Yes, stomata. Stoma. Okay, ground tissues have 3 types of ground tissues. So now you need to know. The first one is what type of ground tissues?
Saima.
Yes, correct. Parenchyma. Wow, with a big nucleus. Okay. How about the center one?
Colonzyma.
Yes, without any nucleus. Without any vacuoles. This one, the parenchyma with a big vacuoles. This one, no vacuoles. How about this one? Cannot see any nucleus.
Sclerenchyma.
Yes, parenchyma. Okay, for vascular tissues, there are 2 types, which is the xylem and the phloem. So the left-hand side is what? Right-hand side is what?
Xylem.
Yes, xylem. The right-hand side?
Phloem.
Phloem. Yes, correct. Okay. Oh. Okay, for the tissue types, we have dermal tissues. So the kenapa ni? Oh, I dah salah buat lah. It should be fade out, then it be fade in now. Okay, never mind. So the dermal tissues have the outer covering protections. They have epidermis, which they are primary growth plant in the cuticles. So we have buds in secondary growth in the woody plants. So we have ground tissues, which can basically carry out the basic metabolite functions, store foods and waters. So as I mentioned, we have 3 types of them. And we have vascular tissues to conduct transport material, conductions and transport materials. So we have xylem, water and dissolved material, whereas phloem is responsible for carbohydrates lah. Okay, because it's going backward already. Just now I do this here. Okay, what is this called? If you want to label, what is this called?
Cortex.
Yes, correct, cortex. How about this one? I think it's this one good. My second.
Vascular cambium.
Yeah, photo, correct. Clever lah, you can score well already lah. Oh, I think the third one I think is here. So what it is called?
Primary xylem.
Primary xylem.
Okay, how about the center one? Let's see later on.
Secondary xylem.
Then how about this one? Anyone want to guess? Okay, this is primary xylem, secondary xylem, you're correct. So this is the cork cambium. Then the outer ring, the most outer layer will be the epidermis lah. I think the second one will be this one. I close it. For about the secondary growth here, what is this called?
Cock.
How about this one? This one
Primary.
Okay.
Primary floor.
Yes, the cork will be the outer layer here. Just now somebody said cork, right? It should be just beneath the epidermis. Okay. Then the primary phloem, just now is the primary xylem. Xylem is the inner. So phloem will be at the outer layer lah. So primary phloem and the secondary phloem here. How about this three? This one I think easier. Primary growth. The purple?
Parenchyma.
The purple one?
Parenchyma.
I don't think so. How about the green one?
Xylem.
Yes. Alright, xylem. How about the purple?
Phloem.
Yes. The center one?
Phloem.
Yes. So they have the selencyma lah, just now you mentioned. The purple one is selencyma. The green, the dark green will be the xylem, and the light green will be the phloem for primary growth. Tutup balik. Tempat I buat salah. Okay. So in the dermal tissues, as I mentioned, so you have epidermis, you have epidermis cell, you have the gut cell. So this is the gut cell. Look more like stomata, but it's gut cell lah in here. Okay. So the gut cell basically consists a pair of stoma lah. Okay. So you have the trichome, which is the epidermal hair, so look like the hair lah. So you have the periderm comprised of the cork cambium. Cork will be at the outer part. Then in between the phloem and the cork will have the cork cambium. Okay. So epidermis cell originating from the photoderm cover all parts of the primary bodies. You have the gut cell. So dumbbell shape is the dumbbell shape. Cell flankings on the stomata. Okay. Control gas diffusions by regulating the opening and closures of the stomata pores lah. You have the trichomes like the hair-like outgrowth, regulate microclimates, and you have the root hair with a tubular extension of the epidermal cell. increase root surface area. So just now I talked about the cork cell already, cork cambium and the cork parenchyma. Then make up the periderms. So this periderm form the outer layer part of the outer stem and roots that cover the plant body in the body plants. Okay? How come like that? Huh? Okay, now you have the open stoma and you have the closed stoma. So this is the conditions. So inner is stoma. This one is what? What is it called?
Gut cell.
Yes, this is called gut cells. So you have the epidermis cell in the outer layer of the gut cells, basically. So this is the trichomes. Outgrowth of the epidermis occurring on shoots, varying well inform and regulate heats and water balance, basically. You have the root hair, okay? Look like the hair of the roots, okay? So we call root hairs. So it's the outgrowth of the epidermis around occurring on the roots to increase the surface area of the substructures, basically. So this is how it look like in the cross sections under the scan. How come? Can you let me do back a little bit? I end the slideshow first. I don't know why. Wait for me. Sorry, yeah. Sorry, yeah, I do wrongly. Yeah. Wait for me for 1 minute. If not, they cannot carry out the games. Okay, let's continue. Sorry about that. I don't know why. Hmm? Sorry, Miss Tawa, how to share back? Now I cannot have the share buttons.
Yeah, at the bottom, right, they got the arrow, share content.
It should have just now, but now only mute, stop video, record. Okay, we continue. Sorry, a little bit technical issues. I choose the wrong command. Okay, so for the ground tissues, as we mentioned, we have 3 types of them: the parenchyma, collenchyma, and the selencyma. So what are the functions of the parenchyma? Anyone want to give me an answers?
Involved in photosynthesis.
Yes, involved in photosynthesis. What else?
Storage for food and
Storage
water.
of food and water.
Yes, correct. Storage food and water. And one more?
Secretion.
Yes, correct. Wow, well done. How about the second one? The coloncyma?
For support.
Yes, for the support of the plant organs. And one more?
Transportation of nutrient.
Yes, correct. Wow, good. Transportation of nutrients. So you all learned before already lah, all these things. How about the third one?
Protection.
Support.
Yes, support
Support.
sections. And one more?
Transfer of water.
For transportation of water, yes. So here we have three types of the cell. So the first type of the cell, what do you think? Which one? 1, 2, 3, which one? This is the first pictures. You think it's parenchyma, collenchyma, or serenchyma?
Parenchyma.
Again?
Parenchyma.
Yes, correct. The first one is the parenchyma. So it consists of intracellular spaces. You can see here. Okay. So they have some rounded shape of the cell. How about the second one?
Collenchyma.
Yes. So you have a collenchyma with the cell wall. Okay. And you have the nucleus inside. How about the third one? You have the stone cell inside.
Sclerenchyma.
Sclerenchyma.
Yes, so sclerenchyma will have the stone cell and the cell wall with lignin. So this is the lignin, and you have the lumen, okay, to form the fiber. Okay, so this is a little bit on the parenchyma tissues. So you have the intercellular airspace. When you look at the cell, you see got intercellular airspace, so it's a parenchyma. So if you arrange like the honeybee cones, then it's collenchyma tissues. If you have the lumens, okay, so you will have the sclerenchyma tissues lah. Anyone can let me know what these are? Oh, sorry, I should use the pointer. This one, the dot, the dark green dot. What
Nucleus.
are they? Yes, correct, nucleus. How about the light yellow?
Vacuole.
Vacuoles, correct. Then you have the light green covering the vacuoles.
Cytoplasm.
Yes, correct, cytoplasm. And one more is the small dot inside. What do you think they are? They are small whitish. Yes, correct. They are start screen. Then what is the line here called?
Cell wall.
Yes, cell wall. Wow, incredibles. Oh, it's correct. Okay. So this is the cell wall. Same. Okay, so you have the thick secondary cell wall, but other features will be the same lah. So this is the sclerenchyma tissues. Okay, in the longitudinal view lah. This is the cross sections. Cross section is when you cut like this and you look at it like this. So a little bit features of the ground tissues. As I mentioned, we have three types of parenchyma tissues: parenchyma, collenchyma, and sclerenchyma. So the shape of it is isodimeric cell, which are oval, sphericals, or polygonal in shapes. Whereas for collenchyma, they have a circular, oval, or polyhedra. And sclerenchyma in variable in shape lah. They consist of fiber and circularies lah. Okay. So nucleus absent in parenchyma lah. And because they are dead tissues, so they are absent of vacuous, absent of intercellular space as well. I think the functions you all know already lah. So occurrence basically parenchyma basically is a packing tissue or soft parts of plant, pitch, cortex, and where parenchyma is a dicot stem, they have in petiole beneath the epidermis lah, absent in monocot and roots. And parenchyma is a dicot epidermis, bundle shaped pericycles in you can see in seeds, bulbs, and fruits lah. Okay. So for vascular tissues, is increase the growth of the plant lah. So it can grow. That's why the plants tree can grow up to 100 meters high. So xylem basically is a dead and lignified and functional maturity. There is a principal water conducting tissues consists of vessels and the trachea. Okay. So we have primary xylem, as I mentioned, derived from procambium of the apical meristem, and we have the secondary xylem derived from the vascular cambium of the lateral meristem, which will have wood made of accumulated secondary xylem. Okay. So let's look at the pictures again. What is this light blue color of?
Primary
Fluid.
phloem.
Light blue, this one.
Primary
Phloem.
phloem.
This is outer part. Xylem will be in the inner
Secondary.
part. So this will be the primary phloem.
Fluid.
Inner will be the primary.
Xylem.
Yes,
Xylem.
correct. Xylem kat dalam, inside. Phloem is outside. Okay. So what is this ring called?
Cortex.
This ring connected in between the xylem and phloem.
Vascular.
Is
Uh,
yes, it's a cambium lah. We can call cambium. Vascular cambium, can. Okay, how about this one? The outer layers, this one, the black color will be the,
Epidermis.
Epidermis.
yes. So beneath the epidermis will be,
Cortex.
yes, correct. So this is the answer. So just now you talk about cambium, yes. Okay. So basically, tricot is a longer, thinner end stepper in many not flowering plants. So the only cell in xylem lah. Whereas the vessels is vital and cylindrical joint at the peroration plates. Okay. So you have phloem. Phloem is the alive functional maturity plant parts, tissues, sorry, muscular tissues. So the principle of fruit conducting tissue in muscular tissue located toward the outer part of the roots and the stem. So carry out the sheath plates, bambos, or we call sheep tubes elements. So you have sheep plates, which is the porous. Some have larger pores called sheep plates. And each sheep tube's member associate with the companion cell and with the nucleus lah. So the cell type, you have the sheath tubes, companion cell, fibers, and phloem parenchyma cell. Okay, look at these pictures. It's a sheath tube bambos. What is this actually?
Sieve plate.
Yes,
Sieve plates. Shift tube.
Yes, shape tubes. This one, the purple one.
Nucleus.
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 So this orange color. So not orange, it's pink color, right? Okay, pink color.
Companion cell.
Companion cell. How about this one?
Sieve plate.
Sieve plate.
Shift plate.
Yes, correct. Oh, got score 100 marks. Huh? Okay, look at the vascular tissues. Aduh, salah apa lagi? We have xylem and phloem. Green color is what?
Xylem.
Vessel.
Xylem. How about the orange color?
Flower.
Phloem.
How about the connector cell between the xylem and the phloem?
Cambium.
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 Yes, you are correct. Okay. Okay, so xylem is one way only lah to differentiate between the phloem. You have water and mineral inside. There are no end walls, and they have a thick wall sheath with lignin lah. Whereas the phloem, they have two-way flow, pergi dan balik lah. Okay, forward and back. Then we have water and food, and the cell have end wall with perforations lah. Okay, so this is a little bit summary of it. So again, you have dermal tissue, ground tissue. With permanent tissue, we have dermal, ground, and vascular. For meristematic tissue, we have apical and lateral cell. Okay, so please remember dermal, ground, and vascular tissues are the primary plant tissue system. Okay, let's do another games with Quizlet Plus. Okay, so I stop sharing first. Okay. The sharing of the code.
No.
Yes or no?
No.
No. Okay. I don't know why. What is the happening?
Yeah, it's just showing your desktop, doctor.
Showing my desktop. Okay. I share again and see. Pause. Okay. Sharing now?
Yes, yes.
Okay. You can scan. I copy the link as well just now. Got some of your friends mentions want to copy links here. Okay. You all can start joining. Okay, we wait 1 more minute, sir, to let everybody join. Then we shall start by 9:51 by my clock. Okay, let's start the games. Okay, this game is by group, so that means you need to, if your group scoring, then you'll be scoring in a group lah. It's a randomized. Okay, let's see who is the winner. Group 1, Putri Christina, you're the winner. So, congratulations. Group 2, Kira. Group 3, Aisha. Group 4, Shakira. Group 5, Ira. Wow, we have many groups. I just last through lah. You can see the winner have the champion here. Group 10, Chen, you have 5. Corrected. Correct. Wow, congratulations. Okay, we have 32 groups of them. Okay. You want the one? I have one more games. Do you still want to play or want to end this class already?
Play.
Sorry again?
Main.
Main lagi? Okay, ada satu lagi. Last. I ada set lagi satu. Main lagi ya. So, quick games. You nak blast ke you nak Quizlet live? Quizlet live will be the first game you play. Blast will be the second game just now you are play.
Main tembak-tembak.
Nak tembak-tembak blast lah. Blast.
Ya, betul.
So tadi dah try this two, the third one this one. Okay, you can start join. Okay, we wait for 1 more minute, sir. So we start by 9:50. Now it's 9:56. We start by 9:57 lah. Mr. I can hear the music of the game, actually. I don't know.
Slightly, Doctor.
Sorry, I just wondering, when I play the game, it got music. So you got the background music of the games before we start? Is it playing? Or you all can hear? Only I can hear. Just to confirm.
It's like slight sound only, but I cannot hear the music.
Oh, okay. Maybe I don't know how to share the sound.
Because normally it have, when we share the screen, on the share screen, they have optimized for the motion and video.
Oh,
So when we click that, so it will show together with the sound.
okay. So share computer audio lah.
No, when doctors can.
Any sound just now? Yes.
Yeah, got.
Ah. Okay, I think the participants stop at 122. Okay, let's start. Great game. Okay, we have about 31 group. Okay, let's start. Okay, congratulations. Okay. Johan wins by shot. Nana by Kira. Okay, that's all from me. Thank you. Again, I pass back my platform to Miss Tiwa. Miss Tawa, sorry.
Thank you, Associate Professor Dr. Tan Suat Hian, for very comprehensive presentation. Dear students, we will now open the floor for any questions. Just a reminder, feel free to drop your questions to the topic through the chat box. Or you can unmute to ask your questions along with your full name and affiliation. When asking a question, please turn on your webcam. Well, since time for Q&A session is 10 to 15 minutes, and due to the time limitation, we are going to select first 3 most voted questions for the first 4-hour speaker. Any further questions after the stipulated time, we highly recommend you to discuss with the presenter via email or through the personal chat. Any questions from the participants? Dear students, please use, this is the good opportunity for you. You can ask Dr. Tan. She is expert in this field. So I hope it will be helpful for your final exam preparation.
Madam,
Yeah,
some tips to study for final.
some tips to study for final. Wow, very good questions. Okay, so I already asked your lecturers. So basically, they will talk about a little bit on the terminologies lah. And some of the things you need to know is how to identify between the cell. So just now I show some of the cell, so I blank it out, I ask you all. So this is how the questions will be. And also a little bit on the labeling. So the activities also cover on that lah. So basically, usually the questions will be like that lah. Okay, do we have to memorize the structures of the cell wall? Maybe Mr. Wan need to answer codes about I not the creator of the questions.
Okay, Dr. Sure. So I also would like to share about the tips that, because since we are teaching BIO122 since many semesters and years, so I hope this will be also great sharings to the students regarding the preparation of the final exam. So generally for the plant tissue topic, so you should know about the structure and the function of the plant tissue types. For example, there are three main types of the tissue, right? So you should remember how to draw and label the structure of the plant tissue types, including their cells, their function, and their characteristics. Because based on the lesson outcome, these are the one mainly you should remember for this topic. So when you do your past year questions, so before this, come out regarding the ground tissue system. Okay, then what other topics come out for this plant tissue? The comparison between parenchyma, collenchyma, sclerenchyma. It's also one of the popular questions. And then the function. Maybe the questions they can also ask in terms of the function. They can ask you to compare the tissue types based on their function. So you must remember the drawing, the label, their characteristics, and the function. So of course, for this topic, you should remember their structure. So try to draw and recall back the structure. That is very, very important for this chapter, this topic. Okay. Then other than that, the dermal, vascular, and also ground tissue. Comparison between these three types of the tissue also previously already come out many times. Then the comparison between the three types of the ground tissues also already come up. So you must pay attention on the questions. They can ask in terms of the structure or the function. Okay, so if they ask about the structure, you have to explain in terms of their structure. So if let's say you provide the answer in terms of the function, you won't get the marks. So you must pay attention. What is the keyword they're asking about the topic? But generally for this topic, you should remember the drawing, labeling, structure, characteristics, and the functions. So that's all from me, Dr. Tan.
Okay, so I saw one questions about how to differentiate between primary xylem and secondary xylem. So I will share back the slide to Can you see the slide?
Yes, we can.
Okay. So I will not project lah. So if do not project also can see, right? Clear, right? So you can see if you have the primary xylem, so is like same structures like the primary growth in stem. Okay, it's like a nuts like that. So if you have the secondary xylem, it will grow old, so it become a ring type like this. Same for the secondary phloem also, it's a ring type. Then the primary phloems will be like the nuts type lah. Okay? Hope I answers the questions. Or Miss Tawa want to add some more?
Actually, Dr. Here we have other lecturers also. So maybe we can hear from other lecturers. We have Dr. Shazwani, Madam Nur Hazira, Madam Shakila, Madam Nadia Nisha, and also Madam Wan Nurul Hidayah. So any of the lecturers also can provide your tips regarding this topic, because many of the lecturers here, they already taught many years also the Bio 122 subject. So any lecturers from here wanted to share any tips for the students, at least they can use for their preparation before the final exam? Or maybe Dr. Shazwani, behalf of Sabah campus, you can provide your tips also regarding the topic.
Okay, well, actually, Miss Taba lebih expert lah. I also knew apa ni, teach this subject only like 2 semester. So I think my tips is just the students can, you know, download past year, past semesters punya questions from the library website and then give it a go. Try do that as a practice question, and the questions are not going to repeat exactly the same. So if the questions has been asked before, then probably this semester it will be as similar but not exactly the same question. So you have to practice a lot. Try download a few of the past semesters question and try it. Okay, and you can ask your lecturer to verify your answers also. You can come to us anytime if you want to check the answers, because the answer is not provided on the website, only the questions. Yeah.
When will be your final exam, dear students?
Student, ingat tak bila final exam? When is the date of your final exam?
Yes, where is the
Thanks,
date for
Dr.
this subject?
Shazwani.
February.
Now
6
February.
Februari.
Pukul berapa?
6 February,
February.
2:15 p.m.
Yes, jangan tersalah hari ya. Jangan tersalah waktu juga.
Okay, thanks Dr. Nur Shazwani and thanks Dr. Tan for your sharings. I hope it will be helpful for the student. Okay, actually we have another one more session, the game session for the students. So we have the challenging game session in the quizzes. So just for 13 questions, roughly 9 to 10 minutes only. So before I start the game session, actually I wanted to ask the students' preferences. You wanted to do as an individual competition or you wanted to do in the form of the group? So how about yours? Just now, Dr. Tan already provided the individual game and also the collaborative game, right? So which one you all prefer? You feel more interesting? You wanted to, because if let's say 5 or 4 students in a group, so your marks will be together, sum up together for the group competition. Individual marks. So what you all prefer? We have the last
Okay.
session.
So we do lah. You all press 1 for individual, press 2 for group. Senang. Vote now.
Yeah. Press 1 for individual and press 2 for the group, as mentioned by Dr. Tan. So we look for the majority. Okay, doctor, when I see from here, many of them prefer the individual.
Yes.
Okay, so let's we play the game as an individual. So from this, we're going to see the top 3 winners. Okay, maybe it can be from Tapa or the Sabah campus. Okay, so actually I created few links because here we have many participants. So for quizzes, only 100 participants. So that's why I provide the few links. Once you already join, if already full, then you can use the second link to join. Okay, so let me share the QR code and then the link. You all can see the screen. Okay, I will provide the link in the chat box, or you can use the QR code also to join the game. Let me copy this in the. Okay, I have provided the link in the chat box, so you all can refer the link. You directly can use the QR code or the link. Too fast. You can provide your feedback. If you really like this kind of the games, you can provide your feedback. For 95 students are joined. Okay. So still students can join. More than 100 still can join. So we have 321 participants. So make sure all of you join. Confidence is the kind of the reflection of your lecture today. So please stay here for a few minutes after you completed your game session. So we have the photo session and then we will provide the registration link for collaborative teaching. So that registration document is the one will be counted as your attendance for the collaborative lecture of this week. Wait for another 2 minutes. So keep joining. So 441 students already joined. So even though you all share your device, no problem. You can use your mobile phone. You can use your mobile phone to do the game session. So I will wait for another 1 more minute. After that, we start. So make sure everyone you try joining the game session. Last one, then we start the game. Because once I started the game, you cannot join. Good, people, join fast. Okay, so let's restart the game. So are you ready? Can we start? You ready? Can we start? Okay,
Yes.
okay. So challenge yourself, huh? So later we will see who are the top 3 winners for today's Columbic lecture. Okay. Okay, 1, 2, 3, start. Is that students you all can hear the music just now? Can hear the music just now?
Yes, yes, we can hear.
job, Ifam. So fast. Everybody doing the questions. From Nabila, from Pratik. So there are 13 questions. Good job. Some of you answered 9 out of 13. I guess you have another 5 more minutes roughly. Complete your questions. So roughly you have another 3 minutes. Challenge yourself and complete the questions. the questions? Seems like everybody answered the question, right? Anybody still doing the questions? So is that you all can see? Any students still doing the quiz?
Not really clear, Miss Tawa, your sound. Maybe you need to stop the music. Yeah.
Okay, any students still doing the game? So we wait for the last 1 minute, then we end the game. Okay. So from that we check the leaderboard. Who is the top 3 students based on their marks, not the score, because the score you get because of the bonus mark and the power up. Correct or not? Okay. So we wait for another 1 more minute, then we end the game. So good job. Actually, here when I check, right, there are 155 students joined the game. Okay. Okay. So the top 3 students, when I check here based on their marks, Shah 10 marks out of 13, Nabila 9 out of 13, and then Noor Zaironi 8 out of 13. So these 3 students are the winner for today's collaborative teaching. Okay, so give a clap to everyone. So these 3 students from which campus? Noor Zaironi from which campus? Tapa or Sabah? Nabila Shah from which campus? Okay, yes. Zafran, any doubts, anything wanted to ask? Nabila Shah from which campus? Tapa or Sabah? Tapa. Okay, so Nabila from Tapa. How about Shah? Shah from which campus? Tapa. Okay, good. Noor Zaironi from which campus? Tapa. So the 3 winners also from Tapa. But I hope still there are some students, right, from Sabah. Okay, when I check, T-I-I-E, Siti Nur Christina, 7 marks out of 13. Okay, then Mashira Wani, Dia also score 7 out of 13. Okay, so really good job. Give a clap to everyone. Okay, even though we did our collaborative teaching at evening, still all of you joined the session. Thanks a lot for that. Okay. Okay, so these top 3 winners is based on their fast speed and answer with the correct answer. Okay, so today you saw the top 3 winners based on their score, another top 3 winners based on their speed. Okay, so here you can check. This is the overview of all the students' score. Okay, Nur Zaironi. Actually, Nur Zaironi scored 12 out of 13. Okay, Nurzaironi, 12 out of 13. Good job. Alicia, 12. Shah, 11. Okay, this is the overall. Okay, this is the overall marks. Okay, good job. Most of you score 11, 10. So the top, Nurzaironi, Alicia, and then Daniel, Shah. Okay, the rest of the students all are similar marks. 11, 10, 9, okay, 7, 8. Naura also scored 10 out of 13. So this is the overall, the correct questions based on the 13. So far, good job. Everybody also did well. So means I understand that all of you also, you understand the topic. I hope the objective of this session is reached. Okay. Okay. So far, all answered. Good job. Okay. Okay, it's look like we have covered the game session and also the Q&A session about the plant tissue. Great. Okay, so now we come to the end of the session. So before I close this session, I would like to invite our speaker to give us some short closing remark, please.
Okay, thank you for Ms. Tawa again for inviting me for these sections. I hope that you all can get some things from what I have taught and bring back to your final exams. So hope everybody can score well your final exams. Already 10:35. I will not talk long lah. I think everybody also very tired already. So thank you everyone.
Okay, dear students, now we have reached the end of our evening session. We appreciate you being here. I hope this evening session will be meaningful session for all the students. Thank you, everyone. Thanks again to our guest speaker, Associate Professor Dr. Tan Suat Hien, for very informative sharings, and also our lecturers from Tapa and Sabah campus.